Hey guys, so this video is on signal transduction. It's the next video after cell signaling. In this video, we're going to understand the three components of signal transduction, and we're going to understand the difference between hydrophilic and hydrophobic ligands. And by the end of this video, you should be able to use adrenaline as an example of signal transduction. So what is signal transduction? It's the conversion of a signal into a functional change in the cell. In other words, it's converting the message received into an actual response. So there's our message. What do we actually do with that message is called our response. So in signal transduction, there are three main stages. One is reception, which is just receiving the signal. Two is transduction, which is interpreting that signal. And number three is the response. What do we actually do as a result of receiving that signal? So in this video, we're going to break down the three different parts and go through what happens in each stage. So there's just a quick diagram to show you. That's all three stages combined together. You've got reception, our ligand binding to our receptor. You've got signal transduction. There's something changing within the cell. We're interpreting that signal. And our response is the overall response of that cell. So stage one is reception. We know from the last video that we looked at ligands binding to receptors. And so reception is just that ligand actually binding to the receptor. Now, each target cell has specific receptors for specific ligands. And now those receptors are either located inside, which we call intracellular, or outside, which is extracellular of the cell. So this diagram here is an example of an extracellular receptor because it's actually outside of the cell. So why do we have the different two types of receptors? Well, if the ligand is hydrophilic, which means it loves water, it can't easily get inside the cell. So the ligand will actually bind to a receptor which is located on the outside. So you can see in this diagram, this is our ligand or our signaling molecule. It's hydrophilic, so it's going to bind to a receptor on the outside of the cell. It's the exact opposite for intracellular. If the ligand is hydrophobic, in other words, it doesn't like water, it's going to pass easily through the fatty membrane and bind with a receptor inside of the cell. So depending on what type of ligand you have, depends on where it will bind. So hydrophilic is outside, hydrophobic is inside. Stage two is transduction. Now, transduction is different depending on where it bound in the first place. So we looked in the last slide, it was either inside or outside. Well, stage two actually is determined by which way that binds. So if it bound to the outside, it starts activating these secondary messengers which allow the message to pass inside the cell. But if it was an inside or an intracellular receptor, usually that forms a transcription factor which alters gene expression. So it's a little bit tricky, but hopefully these diagrams will sort of clear that up. So here you can see is hydrophilic. So we've bound to the outside and we activate a series of different messengers which alter some part of the inside of the cell. This GIF down the bottom, if you watch, it sort of will show how that works and how the message from the outside is then relayed inside. So here's our ligand binding to the extracellular receptor. All these secondary messages get activated. They then pass the message to all these different proteins and enzymes, the actual specific ones you don't need to know. And you can see that then passes the message inside of the cell, ready to do the next thing. With a hydrophobic ligand, it's going to pass directly through the membrane. It's going to bind with this receptor, which forms a transcription factor. Now that then goes to the DNA and will alter some part of the DNA. So the main difference is if it's hydrophilic, we're activating these secondary messengers that relay the information, whereas if it's hydrophobic, it goes straight in, forms a transcription factor, and then will move to the nucleus and the DNA. And lastly is our response. Now this is the overall result of the signal. So this could be, for example, we want to change a specific protein shape, or we want to destroy a particular organelle, or we want to turn a certain enzyme on, or we want to turn genes on or off. So that's the overall result of the cell due to the message. Now we use these messages for a wide range of reasons. So there are a wide range of responses that a cell might enact as well. So I know it's a little bit tricky, but hopefully this video here, this is adrenaline, which we know is just a hormone that our body releases. In this instance, here's our adrenaline, and it's going to bind to a receptor. We'll watch that here. So first stage one is reception. Now, in this instance, it would be extracellular because our receptor is located on the outside of the cell, up here. We then have secondary messengers, which keep activating a whole wide range of enzymes within the cell. And eventually, that enzyme is going to break down glycogen into smaller sugars. That would be the overall response. So we had reception, transduction were these activating of secondary messengers, and our overall response was to break down glycogen. Now, we would do that in real life if, for example, we needed adrenaline before we're doing a bungee jump, let's say. Our body would enact this process to make sure we had enough energy ready for that flight or fight response. So this is an example of extracellular reception, and therefore extracellular signal transduction. We could also have an example where the ligand passed directly through and bound to a transcription factor. So in review, there are 3 stages. It's either reception, which is either intracellular or extracellular, and that depends on the type of ligand. You've got transduction, which is our intermediate stage, where you've got extracellular ligands use secondary messages, whereas intracellular ligands create transcription factors. And lastly is our response, the overall response of the cell. So there are 3 main parts of signal transduction. Hope this video helped. Thanks.